Hi everyone, welcome to the second episode of Microscopy. This episode is more practical based, we go through a lot of steps. If you haven't seen the previous episode, go check it out, that's more math skills. Okay, so even before we start looking at the specimen, we have to know how to prepare the slide, and we looked at that last episode, and the slide is a part of the microscope. So firstly, we add a drop of water to the middle of the clean slide. Then we cut an onion and separate it into layers. We use tweezers to peel off some of the epidermal tissue from the bottom of one of the layers. Place the epidermal tissue onto the water on the slide. Add a drop of iodine. Now, iodine solution is a stain. Stains are used to highlight objects in a cell by adding colour to them. And lastly, place a cover slip on top. This is a square of thin transparent plastic or glass. And now we're going to look at the tips for positioning this cover slip. Okay, so firstly, we stand the cover slip upright on the slide next to the water droplet. Then we carefully tilt and lower so it covers the specimen completely. And the important thing is not to get air bubbles in there, otherwise they'll obstruct your view of the specimen. Okay, so now we're going to look at how to use the microscope now. So we're going to look at this in two sets of three to make it easy to remember, because we don't really know why, but the brain can remember 3 steps easier. So if we take this in 2 steps, then we're going to remember it more effectively. So step 1, clip the prepared slide onto the stage. 2, select the lowest power lens. 3, use coarse adjustment knob to move the stage below the objective lens. Okay, that's the first 3. Next 3, use the coarse adjustment knob to move the stage downwards until the image is roughly in focus. And if you're kind of unsure about where the coarse adjustment knob is, you're going to have to check out the last episode, and we cover all the different areas of the microscope. Adjust focus with fine adjustment knob until clear image forms. This will improve the resolution of the image, whereas a coarse adjustment knob will improve the magnification. If needed, swap to a higher powered objective lens to see in greater magnification. And you'd repeat all these steps again. Okay, so I have seen this before, and it seems a bit silly because it seems just like some year 7 classwork, but it's before it's asked you to critique a student's drawing. So the first point you need to look out for is drawing use a pencil with a sharp point. You don't want any like thick lines or like blurry lines, it just looks untidy and unprofessional. Drawing must take up at least half the space available. You don't want to cram a tiny little drawing, even if it's really detailed, into the space provided, because you want to make the annotations as clear as possible. Clear unbroken lines. We're not drawing just doodles, these are actual cells, so we need to make sure that we are including the full scope of the cell membrane to clarify that these are actual cells. No colouring or shading, that will just obstruct the drawing. Subcellular structures must be drawn in proportion. There's no point in drawing a nucleus that's bigger than the actual cell, because that's scientifically inaccurate. Include a title and magnification observed underneath, or just anywhere on the drawing. Label important features using straight, uncrossed lines. So you don't want any squiggly ones, because that could be misinterpreted as parts of the cell. Okay, so I'm gonna lastly finish with some exam questions. A light microscope can be used to observe a layer of onion cells on a slide. Describe how you would use a light microscope to view onion cells. Include how you would prepare the slide. So these are all the steps that we've just covered, and this is why it's so important, because it's worth 6 marks. Pause the video and write your answer down now. Okay, so by the way, this is taken from CGP. If you wanted to have some extra practice, those books are really good. This is like the general guidance here for looking at the level of writing. So level 1 or level 0 is really brief. You would only include 1 or 2 points, and then all the way up to level 3, which is quite a detailed explanation. So you do not in any respect need to include all of these points. It's just a general guide. If you are including up to like 3 out of each section, then that would be full marks. Or I just push it to prove to the examiner that you can get those 5 or 6 marks, because there's no issue if you include more, but don't think that you have to include every single point. Okay, that's a short and sweet episode. Thank you for watching. Subscribe if you liked. This is a new channel, so we don't have many videos yet, but if you keep your eye out, then you'll be seeing some more. Okay, thank you.